Hello everyone, welcome to Smart Guru Learning. In today's video, we will take a look at gluconeogenesis, which is a process through which pyruvate is converted to glucose. Gluconeogenesis is a metabolic pathway that generates glucose from non-carbohydrate sources such as lactate, amino acids, and glycerol. Mostly, these substrates or precursors are first converted to pyruvate before entering the main gluconeogenic pathway. For instance, lactate is converted to pyruvate through Cori cycle, which we will discuss in another video. Lactate is the major source of pyruvate for gluconeogenesis because 1. It is produced in abundance through anaerobic respiration of skeletal muscles, especially during exercise, and red blood cells because it lacks mitochondria, because of which aerobic respiration is not possible. 2. Lactate has the ability to rapidly convert to pyruvate during anaerobic metabolism through lactate dehydrogenase. Lactate is efficiently recycled through the Cori cycle. 4. The conversion of lactate to pyruvate prevents lactate acidosis. The liver prevents lactate accumulation and helps to maintain the blood pH balance. Also, some amino acids like alanine, serine, glycine, and cysteine are converted to pyruvate, which can further be used for gluconeogenesis. These amino acids are called glucogenic amino acids. Gluconeogenesis occurs during such situations like fasting or starvation, intense or prolonged exercise, low carbohydrate or ketogenic diets, stress or illness. In all these situations, there is a decline in the blood glucose level. This triggers gluconeogenesis to stabilize the blood sugar levels in the body. This stabilization of the glucose levels is very important because glucose is required for numerous important physiological functions. For instance, the brain, red blood cells, retina, and kidney medulla rely heavily on glucose for energy. Gluconeogenesis mainly occurs in the liver and the kidneys. Gluconeogenesis occurs in the liver during short-term fasting or overnight fasting, low carbohydrate diets, and during stress or illness. During stress or illness, hormones like cortisol and glucagon stimulate gluconeogenesis. It also occurs during prolonged exercise. So, the liver is the first responder and the main site where gluconeogenesis takes place when the blood glucose level declines. Gluconeogenesis occurs in the kidney when there is a prolonged fasting or starvation, generally more than 48 hours. Long-term ketogenic or low-carbohydrate diets, and during acidosis. So, when these conditions are prolonged, the kidneys are activated along with the liver to support gluconeogenesis. This reduces the overworkload on the liver. Kidneys also help to maintain blood buffer pH by producing glucose and consuming protons. The renal cortex of the kidney contains enzymes that help to perform gluconeogenesis and release the glucose into the bloodstream. Gluconeogenesis is stimulated by hormones like glucagon, epinephrine, and cortisol and is inhibited by hormones like insulin. Other factors that affect the stimulation or inhibition of gluconeogenesis are intracellular signaling, metabolic signals, transcriptional factors, etc. If we compare glycolysis and gluconeogenesis, we see that they are essentially opposite metabolic pathways. Glycolysis converts glucose into pyruvate, producing energy in the form of ATP and NADH. In contrast, gluconeogenesis converts pyruvate and other substrates back into glucose, consuming energy in the form of ATP and GTP. Before understanding the gluconeogenesis pathway, let us first briefly take a look at glycolysis. First, glucose is converted to glucose 6-phosphate with the addition of a phosphate group from the ATP. It is then isomerized to form fructose 6-phosphate and is further phosphorylated by ATP and forms fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is then broken into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by enzyme called aldolase. Dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. With this, there are 2 molecules of glyceraldehyde 3-phosphate. Next, 2 molecules of glyceraldehyde 3-phosphate are converted to 2 molecules of 1,3-bisphosphoglycerate through glyceraldehyde 3-phosphate dehydrogenase, and this reduces NAD+ to NADH. Then, 1,3-bisphosphoglycerate is dephosphorylated as one of the phosphate groups is removed to form ATP. This produces 3-phosphoglycerate, which is then isomerized to 2-phosphoglycerate. It is then dehydrated with the removal of water and forms phosphoenol pyruvate. The phosphate groups from this 2-phosphoenol pyruvate are then removed to form 2 ATP from 2 ADP, and with this, 2 molecules of pyruvate are formed. In this way, 1 molecule of glucose produces 2 molecules of pyruvate. Now, gluconeogenesis is the opposite metabolic pathway compared to glycolysis because in gluconeogenesis, 2 molecules of pyruvate are converted to 1 molecule of glucose. Some of the steps in glycolysis have greater negative delta G value, and hence, these reactions are strongly energetically favorable in one direction. Therefore, some of the steps in gluconeogenesis are irreversible, and so during gluconeogenesis, different enzymes or reactions are required to overcome this. Pyruvate to phosphoenol pyruvate is an irreversible step. Therefore, pyruvate is first converted to oxaloacetate and then to phosphoenol pyruvate. Pyruvate is converted to oxaloacetate with the addition of carbon dioxide and ATP. This occurs in the mitochondria. Oxaloacetate is then converted to phosphoenol pyruvate with the addition of 1 GDP and the removal of carbon dioxide. Phosphoenol pyruvate is then converted to 2-phosphoglycerate by enolase, which is the same enzyme used in the glycolysis. Conversion of 2-phosphoglycerate to 3-phosphoglycerate is also a reversible step which occurs through phosphoglycerate mutase. 2 molecules of 3-phosphoglycerate are converted to 2 molecules of 1,3-bisphosphoglycerate as they are phosphorylated by 2 molecules of ATP. Step 6. 1,3-bisphosphoglycerate is then converted to glyceraldehyde 3-phosphate through oxidation of NADH to NAD+. Step 7. Glyceraldehyde 3-phosphate is then converted to dihydroxyacetone phosphate. Step 8. It is then converted to fructose 1,6-bisphosphate through aldolase enzyme. Step 9. Fructose 1,6-bisphosphatase then converts fructose 1,6-bisphosphate to fructose 6-phosphate. The phosphate group is removed in this step. Step 10. Fructose 6-phosphate is then isomerized to glucose 6-phosphate through phosphoglucose isomerase. Step 11. Glucose 6-phosphatase then converts glucose 6-phosphate to glucose with the removal of the phosphate group, and this step takes place in the endoplasmic reticulum. With this, there is a use of 4 ATP, 2 GTP and 2 NADH molecules. Here is the overview of gluconeogenesis along with its comparison with glycolysis. If you have any further questions, then please write in the comment section, and thank you so much for watching, and do not forget to like, share and subscribe to this channel for more videos like this. Thank you so much.